Hello, and welcome to the So You Want to Be a Marine Biologist podcast, where we learn about the ocean, share sea stories, and explore ocean careers. I'm your host, Kara Muzia. Let's dive into today's episode. Hey, do you want to help the oceans? Have you considered a career in marine biology, but maybe just aren't sure where to start? Head on over to my website, marinebio.life, and subscribe to my newsletter. When you subscribe, you'll receive a free PDF download where you'll learn the 7 steps to becoming a marine biologist without the degree. Hello, mermaids, pirates, ocean lovers, and land lubbers. Welcome to today's show. Question: what do turtles do on their birthday? They celebrate. What do you call a turtle that is only awake at night? A nocturtle. My guest today is Chris Figgener, sea turtle biologist, science communicator, and the scientist that filmed the sea turtle straw video that went viral and sparked a movement around the world. In today's episode, we chat about what brought Chris from Germany to study sea turtles in Costa Rica, why she prefers sea turtle research over whale research, and how one seemingly small action can create an entire movement. Please enjoy. Chris, welcome to the So You Want to Be a Marine Biologist podcast. I am really excited to have you on and chat with you today.
Yeah, thank you so much for having me. I'm really excited.
I mean, you've kind of instigated a whole movement against straws, and you've done a lot of really amazing sea turtle work, but I read that you actually, growing up when you were little, were afraid of the ocean, and this is very fascinating to me. So how do you go from being afraid of the ocean to that's what you do?
Well, I recommend having a very stern German engineer as a dad, first step, that didn't want to put up with your temper tantrums in Greece because he was too embarrassed of you making a scene in front of everyone at the beach. And then him going up to the concession stands to buy a pair of diving goggles because you were making that scene and telling you to please stop doing that and just have a look at what's there. There's nothing to be scared of. So that is pretty much how it started. I mean, I don't really remember much. I just remember I was standing at like the water. I was like 2 or 3 years old and there was stuff under the water and it creeped me out. And yeah, after I had the swimming goggles, I could actually really see what it was. And I just thought that was like the coolest stuff ever. And from that moment on, I was really just a water rat, as we would say in German, Wasserratte. And my parents had to drag me out and then would place me also into swimming lessons and all kinds of other stuff. I was a lifeguard for many years. So I really like the water by now. But yeah, it was definitely not like hey, I popped out of my mom's uterus and I was in love with the ocean. So it took about 2 or 3 years.
That's still a very young age to fall in love with the ocean. But I love that's such an engineer for you. Like, there's nothing to be afraid of. I will very logically buy you something so you can see what's underneath, and you will see for yourself that there's nothing to be afraid of.
Yeah, that is exactly, yes. And also very German of him being embarrassed for what his 2-year-old kid is doing, right? Because, I mean, it's a kid, God. But no, it's just like, oh my God, she's embarrassing me.
That's amazing. So how did you get into marine biology, though? I mean, you say you went to the beach and you were a lifeguard, but I mean, really, Germany, you don't really think ocean when you think Germany at all.
Hey, come on, we have 2 coastlines. We do have the North Sea and we do have a coast at the Baltic Sea as well. Just that as a side note. But yeah, no, I think you're right. I mean, it's not like the country that you would say like, wow, okay. And plus I grew up completely landlocked. So I grew up in an area that was known for coal mining, really close to the borders with the Netherlands. And interestingly, it was actually closer for us to go to the coast in the Netherlands than it was to go up to the north to our own coastlines. But luckily, my parents were big ocean lovers, so most of our vacations actually happened at the ocean. And then I think my dad, once he noticed that I was really into the ocean, he started fostering my interest. So he would, give me those documentaries of Hans Haas, which is like the Austrian Jacques Cousteau and his wife to watch. And then Jacques Cousteau himself, of course, he also himself had actually quite a few books of Jacques Cousteau, by Jacques Cousteau and Hans Sass. And when I was 13, I actually started interning at the local aquarium. So they had sea lions and dolphins and all of that jazz. I mean, that's a debate if that's great or not. But what it helped me was really to be exposed to also the scientific side. So there was a lot of marine biologists, behavioral biologists that were doing, their master studies, bachelor thesis, and all of that. And so I think I got a really good idea of what it would be like to be a marine biologist and what I needed to do to get there. And so from that point onwards, all of my decisions in my education as well, because in Germany we have English as a first foreign language in fifth grade, or back then it was fifth grade. And then in seventh grade, you would have to choose your second foreign language, which could be either French or Latin. And I chose Latin because back in the days, it was still a requirement for all the sciences to have your grade Latinum. Total waste of my time. I had to literally sit through 7 years of Latin just to find out that this was not a requirement anymore by the time I started studying. But hey, shit happens. And then I also got to know that, of course, most of the literature is in English, right? I mean, we are right now also communicating in English. English is not my first language. And I did not want to struggle or have to struggle throughout my studies with just, with the language barrier. And so when I was 16, I went for an exchange here to the United States to kind of tackle my English and get ready to, dive into the biology literature in English. So I think there were little stepping stones towards my degree, and then I just started studying just as normal. It's a biology degree in Germany. There's no specialization in your bachelor's up until you get to your master's. That's where you're literally able to choose your courses. Everything else is exactly the same over all the universities. It's just that some universities have a better reputation maybe than others.
Yeah, I love that you're highlighting that it's not a specialized degree in marine biology when you're in Europe, or because my undergrad degree is just straight up biology as well. Like I was able to, because of where I went to school, I was able to take a lot of specialized marine related classes. But for the most part, it's just for your undergrad, you just get a biology degree, and it does make you competitive for jobs. And that's something I get asked about all the time. Like, where can I go for a good marine biology school? And the answer is, if you're just getting your undergrad, just get environmental science, bachelor's in biology, and go from there. And get you volunteered at the aquarium. That probably opened a lot more doors and gave you more experience than your degree did, right?
Oh yeah, totally. And I kept on volunteering and kind of going a little bit above and beyond, I think, throughout my entire career.
Mm-hmm.
So, and I think what was interesting is that I really hated my undergrad. I really did not like it because I felt I was doing so much more other stuff than biology. So, where there was so much physics, so much math, so much chemistry. I did so much chemistry, oh Lord. And then, there was also, of course, biology fields that I wasn't super interested in. So I don't want to lie. I didn't particularly enjoy microbiology or cell biology or even botany. And so I kind of, was a little bit frustrated, I think, at the end of my degree. And it was just thanks to a new professor that offered a tropical marine ecology class in Egypt, which was a big lecture beforehand and a seminar, and then a several month trip to Egypt. um, that I was really kind of rediscovering my passion, because this class was actually meant for graduate students. But since he was so new at the university, he started advertising it super late. And so I think it was about 3 undergrads that had the chance to tag along, and I was one of them. And it was literally reaffirming everything that I've ever dreamed of what biology and being a marine biologist could be. Because we were developing our own little studies in Egypt, collecting data, presenting the data, writing a report in the style of a scientific article. So it was super helpful to really, to kind of understand again of what I was doing, what I've learned, of how I can apply it. And then, the next step was actually that so in Germany back in the days, you couldn't stop after your bachelor's degree. So the actual degree you would get after your master's, it was called a diploma, like a German diploma, but it's pretty much the equivalent of a master's. And there was no degree in between. And I always really wanted to go to Hamburg, because the reason I wanted to become a marine biologist was I wanted to study humpback whales, so cetaceans, especially humpback whale song. And Hamburg, University of Hamburg, has a pretty well-known working group for marine mammals. But they didn't want my degree anymore, so they didn't accept me. And I had to rethink again of, okay, how am I getting to where I want to be? So I really wanted something that was behavior related. I didn't want to have a pure marine biology degree, because I wasn't super interested in all the plankton and the physical oceanography part and all of that. I really wanted to have more something that was animal ecology slash behavioral ecology slash behavioral physiology. That's how I envisioned it. And so I decided to not go to the coast and actually stay landlocked and go to university that specialized on tropical ecology and animal ecology and behavioral physiology. And that's where I got my master's degree in. And because I just told myself, hey, the concepts are pretty much the same across the board. And where I later on will apply those things, that's totally up to me. And I mean, luckily, my calculation kind of worked out.
Yeah.
And I also, during that time that I was doing my master's, I was again able to volunteer. That was kind of what set me up eventually.
Yeah. That's awesome. So wait, where did you go? You went to your undergrad and your master's in the same place. Is that correct? And where was that?
No, so undergrad was at Tübingen University, which is a small town in the south, close to Stuttgart in Baden-Württemberg. That's like the state. And then for my master's, I went to Würzburg, which is part of Bavaria, but it's actually closer to Frankfurt.
Now, did you know after you got your master's that you wanted to continue on to get your PhD, or did you take some time and do other things?
Yeah, I did not want to do a PhD. Actually, in fact, I hated the whole brainwashing that was happening. The whole brainwashing that was happening of making you believe there is no job out there for you unless you have a PhD. And so everybody scrambled to find a PhD position, no matter what the actual topic. And they all hated their freaking guts. I mean, I looked at my friends and a lot of them didn't finish, and some of them finished, but then of course were in such a weird specialized corner in a topic that they would have never chosen themselves. And I was scrambling along. So I decided I don't want any of that. And I said, the world is pretty big. I have a hard time believing that there is no job for me at all with the degree I have. And luckily, I was already connected to Costa Rica at that point and to sea turtles, because I had been volunteering here as a research assistant when I was pretty much just starting my master's. And my back then boss was really interested of hiring me, and that's exactly what he did. So I hadn't even written my thesis, and I just had finished my the data analysis, in the sense of I had just done my lab work. And I took pretty much the raw data with me and moved full time to Costa Rica. I sold everything in Germany, gave everything away, and then I moved to Costa Rica full time and started working here and writing my thesis.
That is incredible. That's like best case scenario, right? You have a job before you actually even finish your schooling. So you said you knew him beforehand. So how did you get connected with this place in Costa Rica? And where was this? Was this at, I might butcher the name, Nāmaka Conservation Science? Hey, Kara here. I hear it all the time. How do I become a marine biologist? Or my niece wants to help the ocean. Where does she even start? I hear from students, yes, and also nurses, HR professionals, and other mid-career folks who wonder if they could still make their dream of working with sea turtles or sharks come true, wondering if it's too late to answer the ocean's call. Here's what I've learned after 20 years in the field and hundreds of interviews. There's no straight line, no magic degree, and no gatekeeper. You create the path forward. If this sounds overwhelming and vague, I've created 2 tools to help you stop wondering what if and start moving forward with clarity and purpose. The first is my free guide, Be a Marine Biologist Without the Degree. Over 1,000 people have already downloaded this guide and taken their first step towards ocean impact. Inside, you'll find 7 concrete, doable actions to help you get started, whether you're just exploring or ready to make a change. Then there's the Ocean Blueprint. This is a 30-day self-paced course that helps you build your unique path into ocean science and conservation without quitting your job or going back to school. Inside the Ocean Blueprint, you'll get daily prompts to explore your interests, skills, and next steps, a structure that turns overwhelm into real momentum, and a bonus section to root your work into ocean connection. You don't have to wait for a degree, a perfect plan, or permission. Whether you're wading in or ready to take the deep dive, these tools are here to help you move from dreaming to doing. The ocean called. Are you ready to answer? Links in the show notes and on my website, karagracemuzia.com. I can't wait to see the waves you'll make.
Well, Nāmaka Conservation Science is actually my own consulting company at this point, so it's a social enterprise. But no, back then it was an organization called ANAI. And this is sometimes, I don't know, call it serendipity, irony of life, but I had literally just stepped the first day my foot into the new university in Würzburg. And, when you're at a new place and you have no clue how to get around, where to go to report to. So I was waiting in front of this professor's office to have a chance to chat with him about his courses and the coursework. And while I was waiting, I looked at the blackboard. Back in the days, people were like putting up paper to announce, for example, I don't know, that they have a flat to rent out or books to sell or, for example, internships as well. And there was this one internship in Costa Rica with leatherback turtles that was actually advertised by a German organization. And it was dated like a few months prior, and I knew, okay, this is too late for this season, but I wrote them, hey, is that something that happens every year or just this season? They were like, yeah, that's something that happens every year, but we are not the ones that are actually, the organization in Costa Rica, and we need to connect you with them. So they did, and I asked them, and they said, yeah, you know what, just apply. And I did, and I got accepted. And so I went for about 5 months to Costa Rica in 2007 and worked as a research assistant here in Gandoca, which is in the Southern Caribbean, right next to the border with Panama. And back then, it was an organization called ANAI. I think they still exist, but they're not involved in sea turtle conservation at all anymore. And that was like my boss back then really liked the way how I worked. It was like jokingly saying, hey, why don't you come back? And I said, well, dude, I still need to write my thesis. And he was like, well, why don't you write it here? And I was like, well, okay, that's an idea. So I found me a professor that would, actually be willing to support me and found me some money because emotionally support and academically, but not necessarily financially. I mean, she gave pretty much her entire lab space and everything I needed, but for the actual travel, I needed to get some money and also for what I was, doing here in the field. And then I went another year to Costa Rica and collected my data for the actual thesis.
That's such a serendipitous story. I love that so much. That it's like you were just looking at a random board, like what's on this, and it was an out of date internship, and you followed up, and it worked out, and it's created this amazing career path for you.
Totally, yeah. And I mean, it also totally took me away from humpback whales, which is funny. Because I was always totally, where other people collected Backstreet Boys articles and copied them, I would literally like go through every magazine, anything, any book, and I would copy out like the articles and all the chapters, and had like this big binders just on humpback whales and cetaceans.
So, do you get to work with humpbacks at all now?
You know what's funny? I have worked with humpback whales in the meantime, actually for quite a few years, because the project in the Southern Caribbean that I, started with and also worked for a few years kind of collapsed. And nothing happened there for, well, almost a decade. And while that happened, I needed to find some other work. And I had been working on both sides of the coast, so Caribbean and then also on the Pacific side. because leatherbacks, that's the species I've been mainly working with, I have been working with, do nest asynchronically. But then I had this one season where I had nothing to do, and I happened to work for a company that was sending volunteers to this one particular project, and they liked how I worked. So I became a research associate with them for many years, and I did part of their humpback whale work. And it is interesting work, but I do not like it as much as sea turtle work because you are always on a boat. I mean, it's not like you with sea turtles, it's such a hands-on experience, such a physical experience. And if you do it right, you don't have to freak out the animal. But when you work with dolphins or whales, a lot of times it's like you're standing for hours on end at a boat looking through binoculars, or if they're really close, you're looking at them. And most of the stuff, the cool stuff, actually happens underwater, but you only see, some fins above water. And you take, of course, a lot of photos of fins and water, and some more fins. And yeah, I don't know. It's interesting, but I kind of miss the walking on the beach, the being able to handle the animal that comes with sea turtle work.
Yes. Let's dive in a little bit more into your sea turtle work. So you mentioned walking on the beach, handling the animal. I know you're on boats with sea turtles as well. So could you describe a little bit what your research is, and maybe we just start like where you started in Costa Rica and looking at the leatherbacks and what that looked like and how that's kind of evolved?
Yeah, so I think in general, there's like two large categories in the work that I do. And I started out in this one category, which is like beach work, as we call it. We have like beach work and in water work. So beach work is usually about the nesting females. So you have the females that come to nest at night, which of course is sometimes very tiring. And you are trying to do or to support or collect data for long-term monitoring studies. So that means you tag those females so you can recapture them in the same season or in the years to come. But it also gives you an idea of how many females are actually present in the population, because all of the females lay more than one nest per season. And then, of course, you're trying to safeguard those females as well as their eggs, try to make or pretty much set them up for success. So that means a lot of times you're relocating the nest to safer spots on the beach where they can incubate without being disturbed by other natural predators or humans that are trying to steal it. Or another big problem we have here in Costa Rica is the erosion of the beach. And if you go to the Pacific side, it's actually temperatures. So they are really, really hot temperatures that are beyond lethal. And so a lot of times we need to shade certain areas in order to incubate the nest successfully. And so for that work, of course, we are on the beach many hours at night, rain or not, and try to do all of that. And then in the end of the nesting season, we are also having a lot of excavations because we need to figure out how successful have we been with our, conservation efforts, because we want to know how many babies we've produced, how successful the single nests were. And so we're digging up pretty much every single nest that has been laid, try to count how many shells we have, how many closed eggs we still have, and are able to, from that data, to pretty much calculate like the overall percentage of hatching success, the overall percentage of emerged babies, and then also get an idea of how many adults we might have.
Measuring reproductive success like tells part of the story, right? You can count how many nesting females are coming up by their tracks and also visually seeing them, and then you go back and then you're digging up these nests so you see how many eggs hatched and didn't hatch, if there's any live or dead turtles left in the nest, and you can kind of get a more complete picture by looking at that. But in Florida, we dig up just a percentage of the nests because that we get lots of nesting turtles. How many nests do you get, and how many are you digging up?
So it depends on the project, right? So we have, when I work with Eastern Pacific leatherbacks, for example, where we have literally only 9 nesting females and a total of 30 nests. So we dig up every single nest in the sense
Okay.
of, for example, here where I live right now and work right now, we have about 150 to 300 nests. We're trying to find every single nest and dig it up. Sometimes we are not able to. So I would say, I think last year we had about 80% that we were able to recover. But if you have, of course, a project such as in Austria, where we're working with synchronized mass nestings, where you have every month close to like 500,000 turtles on the beach, it's literally like a way of how to even estimate how many there are. So we do transects there, and we are doing also a certain transect and spattered calculation of how many nests were laid and what the success is by having little hoses kind of stuck into the beach before the adibada, and then it's going to be like meter squares that are kind of dug around, so we can calculate of how many nests we have per square meter, how many of them are from the last adibada, how many are from the one previously, and what is the hatching success. And we kind of have to, it's an estimate, so we take a sample of the actual population that's nesting there, if that makes sense.
Yes, it does. So I want to back up a second, and you're describing the eribadas, right? So this is, and you said it so eloquently, a synchronized mass nesting, and it makes it sound like it's a lovely dance, and the turtles are in sync and on harmony, and it's like a beautiful poetic thing. And I mean, I guess in some ways it's very poetic, but it's like kind of chaos on the beach, right? You mentioned how many turtles are coming up on the beach at the same exact time, throwing sand everywhere, and these are not little turtles. These are like a few 100-pound beasts crawling up and throwing sand and making nests. Can you describe what an arabada is like?
Yeah, so it's like an adibada is literally the synchronized landing of, I mean, it depends on the month, but in the biggest adibads, up to 500,000 turtles at the same time on the beach. I mean, at the same time, it's relative because usually the large adibadas happen within 7 days, the smaller ones maybe just 3 or 4 days at night, usually not as much during the day. And you will have at any given time throughout those adibadas like several thousand turtles on the beach, which is really impressive. I mean, they're the smallest turtles, so it's not like you have leatherbacks crawling all over each other, but you have these, 35-kilogram animals. And so I don't know if I would say it's chaotic. It's definitely super impressive still. And I mean, it smells pretty bad because, they dig up each other's nests and also nests from previous adibadas, which like have decaying eggs inside. And it's a lot of noise, which is really funny because they have this little dance in order to compact the sand. And so they are bouncing and kind of hitting the sand with their carapace, and you can hear it. And it's like when thousands of turtles doing that at the same time. It's pretty cool. But of course, you also have to be careful as a researcher if you leave your backpack somewhere unattended, that some of the turtles might happen to, get stuck with their flippers and drag it down the beach and into the water. So it has happened before. Or trying to count some eggs of one turtle and another one either tries to crawl on top of you or is throwing some nasty, disgusting egg mixed with sand goo into your face while you are trying to concentrate and see how many the other turtle is popping out. So it is, it's like this mixture of like being super in awe, but also super disgusting. And you smell absolutely atrocious after a night of Adibada, I can tell you that. But yeah, I mean, I can recommend if you ever have the chance to see it, definitely do so. We have three, like two and a half Adibada nests or Adibada beaches here in Costa Rica, which are really super, super cool.
That's awesome. I have a few friends that have gone down to volunteer to do that, and it's on my list because I've just heard stories and from what you described, and it sounds absolutely incredible and like quite an experience.
Yeah, it is definitely an experience. And the nice thing is you will never have a lack of data or, not enough turtles, which happens if you work with a species like, the Eastern Pacific leatherback, where you can walk at night for many weeks without seeing a single turtle. And then finally you have one. And then you have weeks again without a turtle,
Right.
which makes you appreciate, turtles that come more frequently, a lot more.
That come regularly. Right. So, how did you decide to get your PhD then? I mean, you got your master's, you had a job. Like, how did you make that jump?
Yeah, it was okay. So I was within this, like, working on both coastlines from actually several years before I got a little bit frustrated maybe with, me being a female scientist in a foreign country, having a lot of male bosses, that were just in the office, not really having the insights of field work, but making the final decisions on a lot of stuff that I didn't always agree with. Also, they were blocking sometimes my trial to publish any scientific data. Since it wasn't my data, I couldn't really go without them. And then lastly, the last straw was actually that one of my good friends and colleagues was killed here in Costa Rica at a nesting beach on the Caribbean, just close to the big shipping port that we have here. And I think it made me feel very acutely that maybe my impact isn't big enough. And I felt very powerless. And I think I just tried to figure out a way of how I could increase my impact, how I could do more, how I could, put the lever at a different point. And so the only way of how I could think about it was like, maybe I need to go back to school to get a PhD. So I would become an autonomous scientist. I wouldn't have to depend on other people signing for me, because I also had a conservation, or I still have a conservation biology class that regularly happened here in Costa Rica. But I always needed, like, other professors that would sign up for the credits because I didn't have a PhD. So all of that was kind of feeling, okay, I want to be an independent scientist. I want to make my own decisions. I want to have my own NGO. And I feel like I need to get my PhD. And that was really the reason of why I set out to look for a lab and somebody that would align with what I was interested in. That was sea turtles and hopefully Costa Rica. And that is how I stumbled across my academic advisor, Dr. Pamela Plotkin, who is at Texas A&M University. And yeah, and it was really great because she knew Costa Rica. She was really passionate about what I was doing as well. And so we had a really great time while I was doing my PhD.
That's awesome. You brought up a lot of points. So the reason why you couldn't publish the work that you were doing is because technically you were working for another organization, right? And so the data that you're collecting was really their data. Is that kind of explain it?
Yes, absolutely. That was exactly the problem. So, there were situations where it was like saying, hey, for example, we had just discovered that we were able to raise the hatching success of leatherback nests at our beach by cleaning the sand with seawater, or actually taking the sand from the shoreline where the water was washing. And we were able to raise our hatching success from 15% to 65%. And I said, hey, we should publish that. And my boss was like, I don't think this is interesting enough. And then I at least was kind of able to present that at a symposium. But then another professor came up to me and was like, so, Chris, are you going to publish that? And I was like, I don't think so. I was told we're not. So he got a student, repeated our, all entire study and published with her, which was totally legit. I mean, he even asked me, but I knew that I wasn't going to be able to publish it, right? Because I wasn't the main boss right there.
Yeah, that's very frustrating. So wait, tell me again what this paper that you didn't get to publish said. The nests were getting too hot, and so that you were able to like sprinkle water on them and cool them down enough to increase their reproductive success.
Yeah. So what happened was that in Austria, where we were working, you have the synchronized mass nesting. So you have an insanely amount of decaying organic matter on that beach because of all the olive ridley nests that are incubating there. So the sand is actually more like soil and not like sand. But also we have the problems with these really high natural sand, like the high sand temperatures. But for many, many years, the project that I worked for was trying to lower the sand temperatures. And by the time I started working there, I was like, well, the temperatures are okay at this point. So the temperature are not the reason that we're not getting beyond 15% hatching success. So I kind of kept on thinking, it's like, well, every time we build the hatchery, we're only sifting the sand. We're not really, getting the organic bacteria, fungi, and whatever else there might be in there out. And of course, they're using up oxygen, and our eggs need oxygen. So I decided, okay, we need probably to put the hatchery slightly closer to the waterline so there is more tidal ventilation. And then we had this idea that, first of all, we did it in secret. We actually didn't tell our boss because we thought he would not agree, to just do one line of the hatchery with, the sand from the waterline. And it was like a total success. So in the end, we presented it and said, hey, I think this works. So he gave us the permission to do half the hatchery. So I decided to have half the hatchery. So you have a control and of course the treated side. And it was so obvious that, the treated side had 65% hatching success, whereas the non-treated side had just 50% hatching success, about the same as always. So after that, we were completely switching to that new technology or the new treatment method. And it was just instead of taking the sand from where it was and sifting, we would literally take out all the sand and about 1 meter, a little bit deeper than that, from the square that we measured, and then fill it with the sand from the waterline that had been completely, washed over with the tide.
Mm-hmm.
So it was a lot of work, but so worth it because it did work. It did make a difference.
That's really interesting. So so you do move the eggs then. You're taking these eggs that are laid and putting them in your own little incubator or hatchery, as you called it.
Yeah, absolutely. I mean, I always am very critical of conservation measurements that are very much messing with natural selection, but certain, populations that are already that critically endangered, such as the Eastern Pacific leatherback, which has about 300 nesting females left from Mexico all the way down to Ecuador. And then we have the situation in Austria now where literally every single natural leatherback nest has a 0% hatching success. So there was enough justification to say, okay, we cannot leave these few nests and try to incubate them because we already know they're not going to incubate. So we are at least going to hatch some babies from those nests in this, controlled environment.
Yes.
But for example, at the beach that I'm working on right now, we are not even having a hatchery. So we are relocating if they're too close to the waterline, if they are in danger of being poached. But other than that, like, I really prefer to leave it natural because, nature usually knows
Right.
best. And you don't know if, a turtle has any kind of defect that might be laying its eggs too close to the waterline. And if she's passing that defect onto its babies, and that way we're creating all little retarded turtles. Yeah.
That's fascinating. And yes, I mean, it makes sense. Let nature do its thing for the most part. But when we're at critical levels, I love that the intervention worked and you found this method that actually really helped in a pretty non-invasive way.
Yeah, it was exhilarating. And I mean, that's just one example of where I wasn't able to publish.
Let's talk about what you did publish.
I haven't actually published that much for the time that I have already worked in sea turtle conservation, for that reason, which is really frustrating.
Yeah.
But I have been able to publish right now my master thesis, which was great. And I have been able to publish at least the first chapter of my PhD already. I'm still having about 3 or 4 papers in the pipeline. But yeah, I can tell you it's a painful process to publish. Gosh.
Why is it so painful? Can you give like an overview? And then I do want to get into some more of your research because you do have quite a few things published. I'm looking at your scientific publication list right now. Why is it so painful?
Why is it so painful? Yeah, it is painful just because I mean, first of all, I think I have gotten better at it, but in the beginning when I started out, just not being native speaker is very, I know it's a lot of imposter syndrome, I think, right there. So I have always felt my English is good enough, to really write at that
Mhm.
level. So I think I a lot of times just kind of procrastinated and didn't do it. But then again, also one of my first big publications was a review. And oh man, I told every one of my friends if I ever, ever, ever have the idea of writing a review again, that they should slap me really, really hard in my face. And yeah, because it's so much work. And I think for like 1.5 years, I haven't done anything else than, looking through different research papers, summarizing that research, and getting frustrated of lack of information or how people have written their paper. And yeah, I mean, luckily it went out eventually. It was a really big chunk of writing, and I'm super proud of it now, but I do not want to repeat the process.
So to bring it down a little bit, so a review is like actually reviewing other people's research and the papers that they've published, and you're making your own scientific and drawing your own scientific conclusions and deductions based on their research versus what you were doing before, which is like going out in the field and collecting your own data and synthesizing your own data, correct?
Exactly, yes. And to make it more interesting, so we didn't only like pretty much gave a summary of like what has been published in the field of stable isotope analyses and sea turtles, which is a way of how you can analyze diet in sea turtles without actually having to look at the stomach content, but you just need to take a snip of a tissue, can be blood, can be carapace, so the keratin layer of the shell, or it can be a piece of skin as well. And you can travel back in time to, for example, when I take it from a nesting female, I know what she ate like 6 months ago, or 2 weeks ago, or even 13 years ago in some cases.
This blows my mind. Genetics was like not my favorite topic. So like this whole stable isotope analysis is like mind blowing to me.
Well, okay. Yeah, it's a really neat technique. It's not super accurate. It becomes more accurate if you're also able to sample potential prey items. And then
Okay.
there's this really cool application, kind of advanced Bayesian statistics, where you can actually say, oh, she probably eats mainly whatever, that's this species of algae or that species of mollusk or whatever. But in the basic sense, you're usually using, in the majority of the cases, two stable isotopes, which is stable carbon and stable nitrogen. And
Okay.
stable isotopes in itself, it's kind of, if you go back in chemistry, you kind of have the same element in with different masses. So you usually carbon is 12, but the stable carbon is 13. So it works the same. It's just that photosynthetically, like certain plants preferably use either carbon 12 or carbon 13. So depending of what the ratio between the two is, you actually know where that plant is growing. So usually, or what photosynthetic process they use. So if you remember, that is like back in high school, we learned about like C3 plants and C4/CAM plants. So usually in the ocean you have C3 plants, and on land you have C4 CAM plants. And that means there is like this, first of all, carbon. If I take a snip from a turtle now, um, it is pretty much the carbon ratio that she has reflects of what the baseline producers are. So that means what are the photosynthesis making organisms in her environment that she is, part of the food chain of.
Mm-hmm.
And carbon therefore gives you an idea of the spatial component. So does she feed close to the coast, or does she feed in the open ocean? So because the more outside in the open ocean is, the more depleted carbon 13 is, or the ratio is, because algae use a different type of photosynthesis than land plants. And if you're close to the coast, you have a lot of river runoffs. So that means you will get the signature of plants that are like C4 and other. And then nitrogen is really cool because nitrogen actually gives you an idea of where in the food chain the organism that you're sampling is based, because there is a certain form of enrichment. So that means if you are a herbivore and you're eating on a plant, so your nitrogen is not the same as the plant that you ate, but it's slightly enriched. And so with every step, so then you are eaten by like the first level predators. So that predator's level is again a little bit enriched in nitrogen. And
Mm-hmm.
so the top predators have these really high levels of nitrogen. So for example, even in humans, if you're vegetarian and I take a snip from you, I can tell you're vegetarian because you're like, not having a very high nitrogen ratio.
Mm-hmm.
And then if I would take a snip, for example, from a meat eater, I would totally be able to tell that this person is eating meat. Um, and if I take, for example, different tissues, I might be telling, hey, but a year ago you were still eating meat, but now you're eating just plants. That's the idea of the tissue. So if I, for example, do layering of the carapace of sea turtles, I can see, for example, because sea turtles usually when they're small, kind of post hatchling, they go out into the open ocean, stay there for several years, and then recruit back to coastal waters. And usually, most sea turtle species, even though they might become more herbivorous as they grow older,
Mm-hmm.
so that means like eating more plants when they grow older, they mostly are omnivorous when they are out in the open ocean. And then you can see this, sharp decline in nitrogen values when they recruit to the coastline. So that means if I layer their carapace and can travel back like several years, a lot of times I can see, oh, when did she actually recruit back to the coast? I can see that she was first an omnivore and now she is a herbivore. So that's kind of the cool thing with stable isotopes.
That is just mind blowing.
Am I smoking your brain right now?
Yes. That is mind-blowing to me. Very cool.
Yeah, going back to the research and the review. So I have done original research on it. So I have sampled, sea turtle populations and different nesting beaches, and I was actually comparing synchronized mass nesting olive ridleys with in the same population we have females that nest solitarily. So I wanted
Mhm.
to see if there is a difference in feeding ecology. And that was my original research throughout my PhD. But in order to kind of lead up and say, like, set the stage, I did this review where I was looking, okay, what does it actually look like in all the other sea turtle species? What has actually been published and what are the findings? So I was summarizing, the findings, but then we did like this massive meta analysis on top of the review where we literally kind of mined data from all of those papers and put them into a huge data set all together and try to reach new conclusions by using all of those data together.
Incredible. And sounds like a lot of work. So I can understand why you felt a little bit shell shocked after your review.
Yeah, it was. And then, of course, a lot of weird math that we needed to do where, we had to invent certain codes in R that were not even, part of a package or other. And I had to consult quite a lot with my statistics professor if, if the route that we were walking on was actually correct. But yeah, eventually
Right.
it worked all out and we got a great publication out of it.
So I want to walk through one of your papers, like one of your original findings, right? So, like soup to nuts, what does it look like? You go out in the field and you're collecting, like, let's say your stable isotope research. Although you have something here where you have the epibiota, right? Is that when you're studying epibiota? So, for listeners, epibiota is living critters that live on other critters. So, like barnacles living on sea turtles would be considered epibiota. Was that one of the things that you're studying isotope analysis on?
You can, but I didn't. And it was actually, I'm just a co-author, so I'm not a first author. And yeah, it was actually the the study that we did when we found the turtle with a straw lodged in its nose.
That's where I was going with that. That's what I thought. You're out in the field, you're collecting data, and then you're bringing it back into the lab. And what does that look? What does that analysis look like? Because a lot of listeners think that, like marine biologists, you're out in the field all the time, and you do get to spend, depending on what you're doing, you can spend a lot of time out in the field. But if you're doing your own research and your own synthesis and writing your own papers, it looks significantly different. Whereas if you're a field tech and you're just out collecting all the research all the time. So could you kind of explain a little bit what that may look like?
Right. Yeah. So, for example, like for my master thesis, I think that's a really neat small project. So I did a what we call a paternity essay. So I was interested
Mhm.
of figuring out what the mating system is for leatherback turtles in the Caribbean. That means I wanted to know if females mate just with, one male or if they maybe mate with more than one male and if those males also might be mating with more than just one of the females, and if I'd be able to detect that. And what I did is I pretty much used like really simple Mendelian genetics. So I think most of us have that in high school where, Mendel kind of figured out that, hey, first of all, we have kind of two sets of DNA. One comes from the dad, one comes from the mom. So that means for eye color, for hair color, we usually have two genes that could, in theory, be expressed. But in our, that's what we call our genotype. But our phenotype might just show, one of the two things. That's like in the most basic form. But that, of course, means that when I sample the moms and then I sample her babies, I know what the mom looks like and I can see the genotype of the babies and I can deduct, okay, if they have two parts at this one particular gene, like two parts, so one needs to come from the mom and I can always say, okay, I can see which one is from the mom because I know what the mom looks like, and then the
Mhm.
other one needs to be from the dad. And so there is, in theory, if this female has mated only with one male, there's only so many combinations that can happen, right? So let's say she is AB in like, or she has the genotype AB at this one particular gene. So, and the dad has CD. So the baby can only be AC, AD, BC, or BD, if that makes sense,
Mhm.
right? So if there's all of a sudden a third or fourth or fifth male gene creeping up that is neither from the mom nor from the other male, then I can deduct, oh, wait a second, there's actually another father that is contributing to that nest. So what it looked like though was that I went out, I took a snip of skin from the mom, then I put her eggs into a hatchery where I exactly knew or like could control when the babies hatched, that I could sample those babies. Then I would go out again and try to find that mom again and again, because I also wanted to know if there is a difference between her nests, because leatherbacks nest on average about 5 to 7 times. So I tried to sample at least two of her nests or more if I was able to. So, people had lists of the female ID. That means their external tags and pit tags and whatever. And they knew, oh, okay, this is one of Chris's females. We need to, keep those eggs in the hatchery and let her know that there is a second nest of that female. And then once the baby hatched, I would sample a certain percentage of the nest. And once I had all those samples, I think it was a total about 20 females and then about 700 to 800 baby samples. I had to export them to Germany, which in itself is already a pain in the butt, because there is a lot of issues with permits, because it's an endangered species. It's called CITES, if you've ever come across it. So you need a special permit to export and import it. And then once it was in the lab, I needed to extract the DNA. I needed to do what we call a PCR. So that means, because I only took like teeny, tiny, tiny snips of the babies. So in order to see anything on a gel, so you can make DNA visible, I needed to amplify the amount of DNA that I had. So I used those techniques called PCR to make more of the DNA that's there. And then I put it on gels and was able to pretty much look at the mom, at the genotype. I could see for each, what we call the loci, for each gene, the two different genes that she had. And then, or for each trait, the two genes. And then I could see, okay, these are the babies, and they also have two genes for that trait. Or for that locus. And then I was able to kind of start deducting, how many dads were per nest per female, and if there was a difference in males as well for the different nests that the female had, if that makes sense. So that was, months of field of lab work after I was in the field.
How many males can they have?
Well, I had, I think, up to 4 different males in the nest, if I remember right. So it was on average usually 2.8 or something like that. Of course, that's not it's not, I mean, it has to be a full male, but statistically it was 2.8, I think, if I remember right. But what was interesting, though, is that it seems to be very much correlated with density. So that means populations that have still, high number of individuals, usually the level of multiple paternity, as we call it, is a lot higher than in populations where there's barely any individuals left. So, of course, that, is very some if you already have small population size, and then the few females that are still around might not be able to find enough males to mate, and maybe that also impacts the the fitness of the babies, because we don't know if she is upgrading, for example, each time. If there's sperm competition, that means the stronger and better sperm is pretty much out competing the sperm that she might have already mated with and might have just produced not as fit babies. So there's a lot of interesting implications of that. That there is definitely a density angle to the whole thing.
Yes. Oh, yeah. So, you're out at night trying, we talked about this earlier, you're walking for weeks, you might find one turtle, or you tag it, and then somebody else finds your turtle and lets you know, right? And then, so you're collecting a tissue sample from mom, which is kind of maybe like a hole punch. And then you're taking a sample from her eggs, and then that's the extent of your field time. Now you're back in the lab and you're analyzing all this, and then you're distilling it down. You're putting some math to it, some stats, maybe writing some R code for it, and then it's published. So it's a longer process than just like going out collecting the data and putting it in a database and saying, voila, look at what I did.
Yeah, and a lot of times it's, there's always this joke that you say when you're in the field, every data line seems like so much. But then once you start analyzing and writing it up, you're kind of like, dude, I should have. Or when you're in the lab, you're thinking, oh man, I collected so much. And then when you're writing up, you're like, oh man, I didn't collect enough.
Yep.
Because it's, sometimes a sampling process of like 2 hours will literally just give you one line of data, like in the Excel sheet. It's one data point.
Right.
So, it's an interesting relationship. So as a recommendation, always collect as much as you can. I mean, without even thinking about it, like if you think you have enough, just go out and collect a little bit more.
Yep. All of it. Even if you're like, that doesn't matter. No, just write it down. You'll thank yourself later.
And be super organized.
Yes. Yeah. Being organized helps. It does with all your data. Otherwise, it's just a mishmash and nobody wants to go through a mishmash of data. That's true.
Now, and keep notes on yourself, lab books and stuff.
Yes. Yeah, that's a good point. So I could definitely talk turtles and nerd out on all your research. And we might have to do another episode where we dive more into it. But I want to talk a little bit more what you're doing now. And I feel like what you're doing now got kicked off by a video that you made during your PhD. So you were the one that recorded the straw video, right? That like sparked this movement around the world that woke people up to the realities of plastic and single uses, and particularly straws, and the devastating impacts it can have on wildlife. What was it like pulling a straw out of a turtle's nose? And did you have any idea that it may have an impact like it did?
Yeah, first of all, no, I did not have any idea of what it would become. And I don't even think, I mean, it was just like this moment where we found the turtle and realized it was a straw. It was just like really, I mean, we have seen plastic in turtles before,
Right.
right? Even though I don't study plastic in sea turtles, it has been this really sad byline ever since I started. And I think we have seen so many cases and so much pain and suffering because of plastic objects. A lot of times it's grocery bags and other, but it was so weird to find a freaking plastic straw in a turtle's nose. It was just like the location, also the object, and how it might have gotten there. I mean, I think we put a lot of thought of like how the hell did like that straw end up in the turtle's nose.
Right.
But I mean, for me, it was really more the moment where usually if I find an animal that is somehow impacted, I am not having the chance of filming it because I'm the one that is, trying to relieve the animal of whatever is making it suffer. And so that moment, I had this visiting researcher on board that was interested in ectobionts. And so after I was done with my team to do all our sampling, he was pretty much just kind of going over the turtles and just like take off all the ectobionts that the turtles had, when he found this funny thing encrusted in its nose, which we thought it was a barnacle, and then for a while we thought it was a tube worm, and then we figured out, man, it's plastic, because one of my local assistants actually bit on it and was like, yeah, this is plastic. And we pulled it out. So I mean, it's just like, yeah, of course, hindsight is always 2020. I think the only thing was really when we were driving back, we were totally not speaking because we were just thinking, oh my God, what just happened? Did that really happen? Right. And then the second was because my camera had died right after we pulled that straw out. I did not know up until what point had I actually been able to film the whole process. So we were for like 3 hours in the dark of if we had even documented of what it was after pulling it out. And luckily it literally had, recorded exactly to the moment where we pulled it out and then it died. So I just didn't have much time to think about it. I just kind of communicated with my professor. I said, hey, I have this video. It is pretty gruesome, but it shows the realities of plastic. Do you mind if I publish it as is? Because I don't have time to like edit it. I have to go to bed because tomorrow we have a boat day, like getting up at 4 o'clock again. She was like, yeah, go for it. Like, this is authentic. Don't worry about it. I'm having totally like, I think that's a good idea. So I just started uploading it on YouTube, being in the middle of the jungle, slow internet. And then I went to bed because I couldn't stand there for the next 8 hours to wait till it was uploaded. And in the morning, I just checked and I saw, okay, it's uploaded. Then, I logged out of my YouTube account and I left for the day for 8 hours on the boat without internet, without any connection to the social media world. And when I came back, like the whole thing had just blown up. Like, my email was like running over and people were contacting us. And when we looked at YouTube and Facebook, it was like no, it wasn't millions in this first, but it was like thousands, which was already very unusual for me. I mean, I think my YouTube channel had like 2 videos before that, and I had like 0 subscribers and or maybe 2 or 3 of my friends, and like, a few tens of clicks or maybe a few hundred clicks, and that was it. So having, I don't know, 40,000 or something like it was insane. And from that, it just totally escalated. Within like a week, it was like millions. All the media started connecting with us, and we were able to talk about plastics, which was incredible. It wasn't just people that come to my projects and I give them like a rant about plastic, but I was able to talk to the whole world pretty much about, man, plastic is an issue. Plastic is also impacting my turtles. We all need to do better with plastics. And yeah, that's kind of how it all happened.
It's crazy. It's crazy that, one little video sparked such a big movement. And I'm thankful that it did, because it did. I mean, your video did wake up a lot of people to the realities of plastic and our impacts on the ocean. So, right now, you are with the Footprint Foundation, and they're working to end plastic. So, it seems pretty fitting that your video kind of led you to this moment. Could you explain a little bit about what Pledge to 2050 is and what you're doing with the Footprint Foundation?
Yeah, so sorry, I need to take like a step back to explain it a little bit better because, for me, being in doing my PhD and being super involved in academics and research, and then also being starting to get involved in this whole
Right.
advocacy thing, I was really enjoying myself because I felt, man, this is a great balance to this really serious, science stuff. And plus, I mean, all the data that we have, it's just not getting beyond the scientific community. And so I was really hoping to be able to really straddle those two things, to continue doing my science and conservation work, but also being able to advocate for sea turtles, against plastic pollution, all of that. And for a while, I wasn't sure if I could do that up until Footprint Foundation came into my life back then, just as the company Footprints, who are an material engineering company that are really trying to disrupt the packaging market and try to provide solutions that especially the large polluters, let's talk about like McDonald's, about Nestle, all of those that always look for excuses of why they can't stop using plastics because there are no solutions. And if there's solutions, they're not scalable or they're too expensive. So they're trying to pretty much eliminate all of those excuses while doing really ethical products. And yeah, trying to make a difference in the world. And they founded Footprint Foundation and asked me if I wanted to be part of it. And so I had this incredible opportunity that I was also able to negotiate that, hey, we know that you have your nonprofit in Costa Rica and your consultancy in Costa Rica. You can continue doing this because we want you to be you. We want you to be authentic. We want you to be, the marine biologist you are, reporting from the front lines of ocean pollution pretty much. But we want to give you the platform and help you build your own platform
Mm-hmm.
to advocate against plastic and help us, of course, to really educate people to make better choices and also businesses to make better choices. And so I ended up at Footprint Foundation and for a year now. And this year, I'm really excited. We just launched for Earth Day what we call the Pledge 2050. And this is a pledge where we're trying to get the Generation Zs involved, of making them understand, okay, we need to do something now. So it's not getting as bad as it is prognosed or predicted for
Mm-hmm.
2050. I mean, there was this, maybe questionable data in one of the articles, in scientific articles that said, hey, by 2050, we will have more plastic than fish by weight in our oceans. And of course, that's very, that's pretty overwhelming, pretty devastating, pretty
Mm-hmm.
horrific. And something needs to happen now. And we're really trying to use the power of social media to get people excited to do something against that, to really make the pledge and stop using plastics as much as possible and keep, pass that message onwards. And we're trying to provide information of how you can go about it and so on. It's just launched, so it's not like we did a slow start, as you would call it. So World Oceans Day is going to be the big launch with a lot of little surprises stuff, which I'm really excited about, which
All
I can't
right.
talk about yet. But yeah, so definitely a thing that everybody could do, helping, my sea turtles in the ocean by using less plastic at home. I think that's a pretty easy, actionable item, if you want to, right? And of course, educate other people. That's always a good thing as well. Educate your parents, educate your grandparents. Try to make them understand that they don't have to be perfect, but just a little thing, little things can help. And we need pretty much everyone to be on deck and help. We don't need, all of the zero wasters that are just like, the top 1%, but we really need the entire world population to do better with
Mhm.
plastics. And we need to find ways of how we can regulate the major polluters. I mean, I'm not going to lie about that. Unless that is going to change, not much is going to change.
Yeah, absolutely. It's funny, like I've worked very hard to phase plastic largely out of my life, and it's once you do it and it becomes a habit, it's very second nature and you don't even think about it. And it takes becoming a habit to make it that easy. But what's the saying? If you 30 days and anything's a habit, right? So just pick one thing and spend the next 30 days working at it, whether it's you forego the plastic water bottle and you have a reusable one and practice filling that up. Makes a difference.
Yeah, and you know what? We never forget our phones. So obviously, we're able and capable of remembering stuff if it's important to us. And so I just think if it's important to you, to make a difference, then you will be able to remember to bring your, I don't know, reusable cutlery or ask for no straw, bring your own grocery bags and do little changes in your household. Because what I did a challenge in 2017, I think
Mm-hmm.
maybe 2018, where I was looking for each month for one thing more because I had already, of course, all the standard things. I did have my reusable coffee mug, I had my reusable water bottle, I had my reusable grocery bags, I had a set of cutlery. Yeah, so that was, and I had shampoo bars at this point already. So I was thinking, okay, what else do I have in my household? Let it be face creams, let it be dishwashing soap, let it be detergent, let it be my clothing hangers. So I got really creative. My sponges that I use, to wash dishes. So what else could what do you use and usually throw away after a while? Toothbrush, toothpaste. What else can I find and find like good alternatives for? And I did that for a year, for 12 months, and just like kept on reducing all of that stuff. And of course, while you're researching different things, all of a sudden there's like this whole world that opens up. It's like, oh my God, yeah, I never thought about why I'm not making my own cleaning
Mm-hmm.
products. It's so easy. And why am I not doing this instead of that? And yeah, if you just put a little bit of effort into it, it's like you can have a huge payout. And it doesn't need to be expensive. Actually, I found that I was saving a lot of money by repurposing stuff and like doing my
Mm-hmm.
stuff at home instead of buying expensive products.
It's amazing the payout that it can be just by switching to your own stuff. I make my own cleaning solution as well, and it's like vinegar and a little bit of rubbing alcohol, which during the pandemic was kind of hard to come by. But it's only a little bit of it, and water, and all of that's very inexpensive.
Baking soda is another staple of mine.
So I want to come back to what you're doing and your nonprofit and your consultancy. So could you say again what your agencies are called? And I have a question for you in regards to them. Kind of describe what you're doing with them, though, please.
Okay, yeah. So I have first a nonprofit that we founded a few years ago, which is actually I'm the only foreigner, which I'm very proud of because not that I am the only one, but actually that is almost no foreigners on it because the idea was after many years of working in Costa Rica that most of the sea turtle conservation projects or conservation in general are still managed by foreign
Mm-hmm.
organizations or they are managed by maybe Costa Rican organizations, but people that are not from the area that they are
Right.
doing something in. And I had been around for some time, and we finally had people that got their first biology degrees and were actually capable and having the education to do something. And I want to empower really the local community and especially the ones that are living with the resources that we're trying to protect. And so this is how COAST, Costa Rican Alliance for Sea Turtle Conservation Science, was born. So I'm not even the president or director. That is actually Ariana, who is the first biologist of this like tiny, teeny, tiny town that has a lot of sea turtles. And most of our research permits are managed over the nonprofit. It's a little bit difficult sometimes to manage the finances as well. And so what I did is I founded also a social enterprise, which is Nāmaka Conservation Science, with which I'm able to like move funds a little bit easier. It's not perfect yet, but yeah, it's just a little bit less slow because sometimes, especially if you're working with funds from outside of the country, there were some problems of getting them into the country and getting the accountability and all of that. And so it's kind of a double whammy. So most of my projects are, run by Coast, but are financed by Namaka, if that makes sense.
Yeah, absolutely.
Yeah, and what we do is we do conservation of sea turtles and research, and our research is pretty much applied research. So we're trying to figure out, okay, what type of data do we need to provide better conservation, to provide better protection for our turtles. And of course, we're doing the beach work that we've already mentioned, and we're also doing in water work. So I do a lot of habitat use studies. That means I want to know how our turtles swim around in our ocean. So what kind of, or what waters do they use? Because of course, not every waters belong to a country. So, you know, beyond a certain mileage, it's actually international waters. And they might even go into the waters of another country. And I try to figure out how the connectivity is, if there are certain migration corridors that need maybe more attention in future, so we have to establish some type of protected area that will protect all the animals that are traveling through that corridor. I'm able to establish collaborations with projects that might be operating in the areas where my turtles are traveling to. But I'm also interested, of course, to map out how the turtles are using Costa Rican waters, so I can make recommendations to the Costa Rican government about how can we extend protected areas. Do we need to establish new protected areas and all of that? Because there's very little data on sea turtles in actually their natural habitat. So we have a bunch of data on nesting beaches because that's an easy data set, because you just need to kind of walk onto the beach, look for the nesting females. But it's a whole entire different way of catching turtles and monitoring turtles in water. And yeah, the satellite transmitting is one way of doing that, but it's also really costly. So one of our satellite transmitters is about $1,000 to $2,500, and that's like the most basic model. So it doesn't take a lot of extra data, because you can also look into diving frequency and dive depth and all of that fancy stuff. But I really am more interested in movement. So it's about 1,000 to 2,500 a pop, and we're trying to get more and more, and also use the tracks and the turtles for our outrage, because turtles, or people, turtles as well maybe, but people are usually excited about turtles. And it is very different if you talk about sea turtles as a whole, or if you have such a video as the one with the plastic straw, where you have the visual of one turtle that is suffering through the procedure of removing it, in close up her face. And the same idea is also like if you have a group of school kids that actually adopt a turtle, they know their turtle, they've seen the turtle when we install the satellite transmitter, and then they can follow her online of how she moves through the oceans. I mean, they have these immediate, emotional connection, because they usually name the turtle, and they start to get really, emotionally involved in its well-being as well, and start to do a lot of cool stuff because they want to help whatever that turtle's name is in the ocean.
Yeah. I love it. I love getting everybody involved. I love the turtle trackers. We have a couple organizations here in Florida that do the same thing, and it is really fun to log on and be like, and where are they now? Oh, they traveled thousands of miles already. Holy cow. They're impressive creatures for sure. So I have a new question I'm introducing to the podcast. If you had unlimited funds that just got funneled into Nāmaka Conservation Science and then subsequently coasts, what would you do with that money, right? If legislation or research, unlimited funds, what would you want to study?
What I wanted to study or what I wanted to do?
Both. Yeah, I guess I shouldn't have said study. What would you want to do if it was unlimited funds, whether it's research or legislation, or what would you want to do with the money?
I would try to get as many early career scientists, especially women, out into the field and be able to like cover their room and board and just be doing cool stuff. And then having, them do all kinds of different projects. I think that would be like, just the equipment for them, everything that they needed to like do a really kick-ass study to get to know what it's like to work as a field biologist. Because, there is still this big divide in conservation biology that it's sometimes regarded as, kind of the rich man's playground. I think also we have a lot of still white males around, and not enough locals, and a lot of, and
Mhm.
at least in Costa Rica, because it's still a very classic gender, very classic gender ideas around. There's not a lot of female scientists, I feel. So I really would like to foster that and do a lot of outreach with kids, and then buy all the satellite transmitters in the world and use all kind of cool cutting edge technology to visualize and do some really cool, yeah, behavioral studies and ecological studies and physiological studies, I think. I mean, hey, gadgets, you can literally, I mean, I could always spend more money. That's not a problem.
I love it. I love that you were like, I want to get all the people out in the field because that's how I got inspired. That's how I got through my undergrad, right? It was that class to Egypt that you were like, okay, this is why I'm doing what I'm doing. So you want to help inspire the others. And then you're like, but I will take all that money and put it on all the turtles and get all the data. That was a perfect scientist answer.
Yeah, that is sick. That would be my dream. And actually, I wouldn't even want to be the first author. I would them to do all the work, which is also easy on me. But it feels so good. Like I have had some situations now ever since I graduated where I was able to provide certain opportunities to people, and you feel like freaking Santa Claus. I mean, it feels so, 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 so good to mentor people and be not just like an emotional mentor, which I've done for many, many years already, but actually to provide some type of financial support at the right moment, that you can see, okay, they have already worked super hard, but this is like definitely giving them the edge and the last bit that they needed to succeed. It feels so, so good. Seriously.
Yes, that's awesome. I love that. All right, another question, and it is my favorite to ask. What is your favorite field story or stories to tell? And this could just be the most epic day in the field. You had amazing turtles or whales, best interactions, or it could just be a day where everything seemed to go wrong, but it makes just such a wonderful story now.
Oh God, I could write a whole book on field anecdotes. Well, we actually joked about that we could either write a telenovela, which would be like very dramatic about all the crazy stuff that nobody would believe, and then it would just be literally one on one of what we have experienced. Or we write like a field guide on poop stories in the field. But maybe I go with most epic. Well, I don't know if it's most epic or not. See, this is difficult. I can't decide. Because literally, I mean, one of the most beautiful things in my line of work is that you have these, net geo moments all the freaking time where you just think, wow, hit me, is that really happening? This is so incredible, beautiful, of course paired with really toxic and horrible moments as well. So I don't know. I mean, I always love nights where you have, for example, here in the Caribbean, where you have like a bunch of turtles and you're so busy that you're just running from one turtle to another, barely able to think, of about yourself, but just think, okay, okay, I need to do this. Okay, how am I positioning my volunteers? So we're getting the eggs. And then you just are totally pooped by the time you leave, such a celebratory mood, because you've just spent your last 4 hours surrounded by turtles, safeguarding eggs. So that's pretty incredible.
Yes, that is cool. It's a cool feeling. Wildlife, just being surrounded by wildlife for hours on end, right? It's a wonderful thing.
Yeah, totally agree. I mean, I wouldn't want to change my life with anybody, to be quite honest.
No, you have a wonderful story, and I love that you're continuing to write it. So, what is a conservation ask that you would like the audience to take away from today's episode that you'd like for them to go forth and do?
Yeah, I think important for me is always to kind of point out that you don't have to come to Costa Rica necessarily to, help. make a difference for my sea turtles, but that can start at home in your everyday life. I mean, like we talked about all the different things already that, might threaten sea turtles. There's plastic pollution, there's climate change, there is overexploitation. So just become, a conscious citizen, try to reduce your emission. I mean, there's a good chance that, if you reduce your animal products that you're eating, that this will help reduce emission as well. That you're reducing your plastic, that will also, produce less plastic and less plastic in the environment. And of course, also, I mean, there is a lot of like projects like mine. We are grassroots, right? So we are a small organization, literally like a 1 or 2 women show, and we don't have the funds such as a WWF or a Sea Turtle Conservancy. And so even small financial support. So we have a Patreon account on Milky Wire. It's not Patreon. It's like a Patreon account, but it's just for conservationists. And it's all small grassroots organizations such as mine. And you can literally start supporting us with like $3 a month. And this makes a huge difference if it's, a few hundred people that do that. So it's not breaking your bank. It would be the same as inviting me to a cup of coffee every month. But for us, it means literally if we're able to continue doing what we're doing the next year or not, if I'm able to pay salaries to my locals or not, which of course is also a double whammy, because if I'm able to employ locals, they will be less likely to go out and poach our turtle nests, right? So it's always a win-win situation. Yeah, so I think that are like, the main things that everybody can do at home.
Yes, absolutely. Milky Wire. I'll put a link to that in the show notes along with everything else that we chat about today. If listeners want to find you, connect with you, where's the best place to do so?
Yeah, I'm pretty active on Instagram, so I think that is a good way. At my handle is Sea Turtle Biologist, very non-imaginative, but yeah, so that's a good one. And of course, email is another good one. I think if you're a little bit Google savvy, you will be easily figure out of how to reach me over email as well. And
Yep.
yeah, but anything like Facebook, Twitter as well.
Your
Instagram's the best.
Instagram is fantastic. I love the nesting leatherback to Eminem. I cried laughing watching that the other day.
Yeah, I mean, I don't know. Do you not have that when you see something that you automatically have a little soundtrack in your head? I always have it. And now with the reels, I can literally put that into like real live music. So you can share on my little private films right now.
Oh, it's fantastic. Guess who's back with a nesting leatherback? Oh, I was like, this is gold. Gold right here. All right. I was going to sign off, but I do have one more question. I used to have what's your favorite sea creature, but somebody asked me that and I couldn't answer it because I realized it changes depending on my mood or the day. So, do you have a favorite species of sea turtle?
Yeah, leatherbacks, hands down. Sorry, all the other species. Yeah.
I feel the same way. Why do you love the leathery beast so much?
They're just the superlative of everything, I feel. They dive the deepest, they travel the farthest, they are the largest. They're like not even real ectotherms anymore because they're able to produce part of their own body heat. I mean, I could just go on and on of how cool leatherbacks are. And it's just none of the other sea turtle species are really able to kind of, measure up with that. It's not. Sorry.
Yep. They're pretty cool animals. I love it. Well, this was really fun chatting with you today. I feel like I could have talked to you about your research for like an entire day. So we might have to do this again. But thank you so much for your time and for being on the show. I really appreciate it.
Yeah, my pleasure. Thanks for having me. It was a lot of fun. I think I haven't nerded out that hard in a while, so that's really cool.
Hey, one more thing. Do you want to dive more into the ocean and marine biology? Need a little guidance on ocean conservation? Head on over to marinebio.life/resources. We've got book recommendations, job posting pages, conference suggestions, and ocean friendly products. All recommendations have been personally vetted by me, and I will continue to add to the collection as I come across cool things to share. Head on over to marinebio.life/resources to learn more. See you over there. Thank you for listening to today's show. I'd love to hear any insight you've gleaned. Leave a comment in the show notes or send me an email over at marinebio.life. If you enjoyed this episode, leave a review and of course, share with your friends. If you want more resources for ocean news, including conservation topics and careers, plus personal insight from me that I just don't share anywhere else, join me at marinebio.life and sign up for email updates. Keep after your dreams and making waves in your community. One person can make a difference. Thank you so much for listening, and I'll catch you next time on the So You Want to Be a Marine Biologist podcast.